All right, today I'm going to go through topic 5, which is DNA replication, transcription, and translation. So basically DNA to protein. I have a little FRQ at the end. It's a short FRQ. I'm mostly relating all of this to gene regulation. I'm going to go over gene regulation a little bit more tomorrow. But what is most important about DNA to protein is the regulation of that process. And so you should have printed out a guide. The guide is going to have a list of vocabulary words and questions. And so the questions that I put on there is what I think is the most important takeaway, because there is a lot of information and a lot of diagrams. I wouldn't get too obsessive about knowing every single step, but just having a general idea of the steps. And then I have a Quizlet link that was in the email where you can go back and study those vocab words. What I'm going to do is just answer these questions, and then I am going to go through some of the vocab, and if you are still confused on a vocab word or you don't remember it, I would jot it down and then seek further clarification on it. So our first question is, what is the Frederick Griffith experiment? So remember, Frederick Griffith was the one that was injecting mice with bacteria. So he injected mice with pathogenic bacteria and they died. He injected mice with non-pathogenic bacteria and they lived. He injected them with killed pathogenic bacteria. They lived. And then he took these two where the mouse lived, right? Mixed them together and the mouse died. And so the conclusion was there is something from, even though this pathogenic bacteria is dead, there's something from it that transformed the living bacteria into something that was pathogenic. So living R bacteria was transformed into deadly S bacteria by an unknown heritable substance. And then Oswald Avery in 1944 discovered that transforming agent was DNA. So that is the Frederick Griffith experiment. So if you need a pause, go ahead and pause. What was the Hershey-Chase experiment? Hershey-Chase experiment was to determine whether the heritable substance was DNA and not protein. Is genetic material DNA and not protein? And so I put a picture here that's not that great, but you'll probably remember that you have a bacteriophage that's infecting a bacteria. This one has been sulfur has been radioactively labeled, and then this one, phosphorus, has been radioactively labeled. Think of it as if something's radioactively labeled, it's almost like it's glowing, they can track it. And they wanted to see what was inside the bacteria after the infection. Was it sulfur or was it phosphorus? And they found that radioactive phosphorus ended up inside the bacteria. So what that means is that the heritable substance was nucleic acids. Why? Because nucleic acids have phosphorus, not sulfur. Proteins have sulfur, not phosphorus. So that's why that was a really great experiment. I'm going to just run through the structure of DNA just to refresh your memory on a couple of things. Remember, DNA are made up of nucleotides, and a nucleotide has 3 parts: a phosphate, a sugar, and a nitrogenous base. The phosphate is very negatively charged, and that is why when we did gel electrophoresis, we load DNA on one end and you turn on a current, and DNA moves from where you loaded it to the positive end of that chamber. And the reason is because DNA is very, very negatively charged because of those phosphate groups. And then we have our nitrogenous bases here, T, A, C, and G. Remember that A is going to hydrogen bond with T, C is going to hydrogen bond with G. And I would just remember A goes with T, C goes with G, and those are hydrogen bonds. The reason that there's hydrogen bonds between our nitrogenous bases is so DNA can be easily pulled apart during replication and during transcription. Two DNA strands run counter to one another. The phosphate group is the 5' end. The hydroxyl group is the 3' end. And the anti-parallel running counter to one another and those negative charges is what's going to cause DNA to twist into that double helix. The other important thing I would remember from this slide is you cannot add a new nucleotide to the 5' end. You can only add a new nucleotide to the 3' end. All right, DNA is wrapped around these little proteins called histones. This is only in eukaryotes, DNA is wrapped around histones. That's how it is packaged and organized. I think that's all I know from this slide. Histones are the proteins that DNA is wrapped around. I would definitely be familiar with the comparison between prokaryotes and eukaryotes, just as a refresher. They both have double-stranded DNA. There's so many times that students will tell me that prokaryotes have single-stranded DNA. That's not true. Both are double-stranded. The difference is that the prokaryote DNA is circular, eukaryotes are linear. Prokaryotes have one chromosome, eukaryotes have more than one. Prokaryote DNA is in cytoplasm, eukaryote DNA is in the nucleus. Eukaryotes have histones, prokaryotes do not. Prokaryotes just have supercoiled DNA, and that's how it's packaged. Eukaryotes have chromatin, and chromatin, remember, is DNA with histones packaged together. So I would just be familiar with the comparison there. And I think for the most part, I mean, we've gone over this and over this and over this, that everyone should probably remember it. If you don't, then I would just kind of focus on it right now and then move on. All right, DNA replication is semi-conservative, meaning that one strand of the parent DNA molecule is conserved in the new strand. And then here are the major steps. So I know that's one of the questions on there. I would paraphrase this the best that you can. Helicase unwinds. Two strands are separated. Primase puts down an RNA primer. DNA polymerase is going to add the new nucleotides to the leading strand. Then you have DNA polymerase that's going to add nucleotides to the lagging strand. Then DNA polymerase 1 is going to replace the primers, and then ligase is going to seal them together. So if you need to pause, go ahead and pause and just kind of paraphrase that. And then this picture just goes through it. Helicase unwinds DNA, and then the initiation proteins are going to separate the two strands. Topoisomerase is going to relieve the strain upstream. Primase is going to add a primer because DNA polymerase can't add new nucleotides without a primer. And then we have DNA polymerase adding to the 3' end to the hydroxyl group. Leading strand is going to continuously go towards the replication fork. Lagging strand is going to go away from the replication fork in spurts as DNA opens up. That is because you can only add, our DNA polymerase can only add to the 3' end, so it has to go in the opposite direction here in spurts. Sorry. And then you have the Okazaki fragments with the lagging strand. DNA polymerase 1 is going to replace primer, ligase is going to seal it all together. And then you have your proteins, your enzymes and their functions. So I went over all of these. So if you need to pause and just read through these, go ahead. I'm pretty sure these are on the Quizlet. What you can do is you can take that Quizlet and you can modify it. If I happen to forget something on there, let me know, or just go ahead and modify it on your own, right? And then there's your final picture. All right, DNA gets proofread by DNA polymerase as it goes, and you have what's called mismatch repair and excision repair. So nucleases are going to cut out damaged DNA, and then DNA polymerase and ligase are going to fill in the gaps. So I want to kind of bring your mind back to that time that we did that sunscreen experiment. It wasn't that great of a lab, but it was okay. UV radiation is going to cause a mutation in nucleases. So if nucleases are mutated, then that means repair to DNA can't happen, which is going to, further exacerbate mutations. And so that was the purpose of that lab that we did. It was fun though. I enjoyed it. I don't know if I'll ever do it again, but I did enjoy it. All right, then we have problems at the 5' end. Okay, I'm just going to kind of skip forward. It says telomeres. What do telomeres have to do with cancer? So telomeres, remember, DNA chromosomes in eukaryotes are linear. So that primer that's added at the very, very end of the linear chromosome cannot be replaced. And so in order to protect us, and the analogy that we used was the aglets on shoestrings. In order to protect us, we have telomeres that are just repeated nonsense sequences of DNA that, hey, it's okay if this doesn't get replaced or it doesn't get replicated, like whatever. So the caps of DNA. So every time a cell divides, or every time DNA gets replicated, these caps get cut off because they can't be replaced. Because remember, DNA polymerase needs a primer, and there's no primer there for DNA to grab onto so they can replace this primer, right? So the telomeres are going to get cut off every time DNA replicates and a cell divides. Once DNA, a chromosome shortens enough, that cell is going to undergo apoptosis. That's a good thing. It's kind of like when a person gets old enough, you're going to take their driver's license away because they're no longer capable of driving and operating the machinery. I don't know if that's a very good analogy, but it's the one I want to use because I don't want to be like morbid with my analogy. So telomeres are good. Telomerase is an enzyme that will come back around and add telomeres back on. So when would we want this to happen? Well, we want it to happen in germ cells, the cells that are found in the ovaries and the testes that undergo meiosis to make eggs and sperm. And the reason for that is you don't want your eggs and sperm to have DNA chromosomes that have been shortened. The bad thing is in cancer cells, telomerase is going to continue to add telomeres back on. So the result is cancer cells are going to be immortal. They're never going to undergo apoptosis. And so the longer that cell exists and keeps dividing, the more likely it's going to be prone to more mistakes in DNA replication. And that's part of the reason that cancer cells are so bad, is that telomerase is actually active in cancer cells, adding those telomeres back on, making it so those cancer cells never get a signal to undergo apoptosis because they're too old. And then this just shows you a picture, just a visual of telomeres and telomerase. Remember, the telomere is a primer that never could be replaced. It's supposed to be removed. Telomerase is going to come in and add DNA back on. All right. And then we move to transcription. So for transcription, explain the step-by-step process. All right, so this is just an overview of, DNA gets replicated before a cell divides. Then DNA gets transcribed into RNA, RNA gets translated into amino acids, and then they fold to make a protein. So the first step of transcription is initiation. That means RNA polymerase is going to bind to DNA before the process gets started. It's got to bind to a very specific point. It's not just going to start transcribing anywhere. It's got to start at a specific point to make sure that it's transcribing completely. It's kind of like you wouldn't pick up a book and go to the very middle of it to start reading the story. You wouldn't get the full story. So RNA polymerase wants the full story. So in bacteria, it's going to bind to a promoter. In eukaryotes, it's a little bit more complicated, of course, because we have more DNA. Things have to be more tightly regulated. There's a TATA box, transcription factors, then RNA polymerase can bind. All right, in both prokaryotes and eukaryotes, you then have elongation. That just means RNA polymerase is going to create a complementary RNA strand to DNA. Complementary meaning, if DNA has a G, RNA is going to have a C. If DNA has a T, RNA is going to have an A. If DNA has an A, RNA is going to have a U. Remember, RNA does not have thymine. So that's the one difference. That's how you can tell you have RNA, not DNA, is that you see U, uracils. All right, RNA polymerase does it all. Doesn't need helicase, doesn't need topoisomerase, doesn't need any of those things. It goes ahead and it just does everything on its own, makes the new RNA transcript. It reaches a terminator sequence where it is signaled to stop, cut the RNA off. In prokaryotes, the mRNA is ready to be translated. In eukaryotes, it's called pre-mRNA and it needs to be processed before it can be translated. So how does mRNA get processed in eukaryotes? This is question 3. RNA gets processed in eukaryotes. This is something really important to know. College Board loves bringing this up. So in prokaryotes, it's ready to be translated. In eukaryotes, it's going to undergo some changes. All right, so what are those changes? So in eukaryotes, it adds a 5' cap and a poly A tail that helps export it from the nucleus and protects it, helps it attach to the ribosomes. It is also going to undergo RNA splicing. That means that introns and exons, introns are cut out, exons are joined together. And remember, I wouldn't memorize anything about this except, oh cool, spliceosomes. They're made up of RNA acting as an enzyme. They're going to catalyze the process of splicing the exons together. So that's part of how RNA gets processed. So I probably went really fast there, but you would just write 5' cap, poly A tail, splicing. Introns get cut out, exons get pasted together, basically glued together. So why have introns? Like, why do this? That seems kind of inefficient, right? Actually, it's not. So we have RNA splicing because we can have alternative RNA splicing, meaning depending on the combination of introns and exons, you can end up with a completely different protein. And that allows us to have 20,000 genes, yet we're able to make 100,000 polypeptides, which is actually pretty cool and more efficient than if we just did it the other way. So I would definitely understand this right here. If you don't, then seek clarification from me or from the internet. All right, now we're back to number 2. Let's go through translation step by step. So you have your mRNA message, your tRNA is the interpreter, your ribosome is the site. All right, tRNA, I always refer back to that little game that we played on the, the computer where you had these little tRNAs and everyone got so inconvenienced because you had to use like the up, down, side to side arrows. So tRNA picked up the little bubbles and brought them back down. I still love that. I'm going to still do it every single year because I think it is the best visual. Hey, it's the best one I can find on the internet. If someone can find something better, let me know. Maybe Labster has something better, but apparently everyone hates Labster. So I'm still going to use that Bioman Bio game with the stupid music. So the anticodon on the tRNA, you see right here, this is the anticodon. It's complementary to the codon of RNA. So this anticodon, AAG, is going to pair with UUC, and it's going to bring in an amino acid. It's going to link the amino acids together. Remember, a ribosome is made of 2 subunits. I wouldn't really get into this, except it's made out of rRNA. And remember, ribosomes are made in the center of the nucleus. Okay, in the nucleolus. All right, there's an A site, a P site, an E site. I don't think I would get too much into this. This is just just like the factory line. Like, this is just the direction that it goes in. So methionine is the initiator. It's a start codon. So you have initiation with methionine. That would be the first step, initiation with methionine. Elongation. Methionine moves to the side, then another tRNA comes in, bringing an amino acid. Those amino acids link together through a covalent peptide bond. Peptide bond between the amino acids. So you see this is your growing polypeptide here. You see how this one moved over, this one brought a new one in, a polypeptide bond formed. There's your growing polypeptide chain. That tRNA leaves and the process goes over and over and over again, and the growing polypeptide chain continues on until a termination or stop codon is reached. So same thing, initiation, elongation, termination. It's kind of the same process. And then you end up with a polypeptide at the end. And of course it's purple. I love how we're consistent with the color of our proteins. If it's purple, it's a protein. All right, and then that long polypeptide is going to enter into a chaperonin and it's going to fold. And it doesn't become, a polypeptide does not become a functioning protein until it has folded. So what is worse, a substitution or a deletion mutation? So remember, mutations happen during replication. If DNA is not replicated correctly, transcription is not correct. So you get a faulty RNA. And if you have a faulty RNA, you have a faulty protein. So remember, we have all these different types of mutations. We have substitutions, insertions, deletions, all that stuff. So which one's worse, a substitution or a deletion? The answer is a deletion, because if you delete one base, everything shifts over and it causes a change in all the amino acids downstream. So I would definitely remember that. A substitution mutation is only going to change one amino acid. And this gives you an example here of a normal situation. This is normal. This shows you when one base gets deleted, how drastic that change ends up being. All right. So that's the answer to number 5. I would also remember that out of all of these amino acids, depending on the property of amino acid, one substitution mutation can be worse than another. So if you have a hydrophobic amino acid originally that was supposed to occur, and the mutation causes a substitution of a new amino acid, but that new amino acid is also hydrophobic, it's not going to be as detrimental as a mutation that causes a hydrophilic to replace a hydrophobic or vice versa. So hopefully you understand what I'm saying there. College Board likes to bring that up too. Some substitution mutations are worse than others because it depends on the properties of the amino acid, the new amino acid that results from the mutation. So hopefully everyone got that. All right, here's a really short FRQ. I would go ahead and pause if you want to try this one on your own, and then I'm going to give you the answer to it. So pause. All right. A researcher is studying the patterns of gene expression in mice. The researcher collected samples from 6 different tissues: liver, heart, brain, kidney, pancreas, and skeletal muscle. And they measured the amount of mRNA from those 6 genes. If it's white, that means no mRNA. If it's gray, moderate amounts of that mRNA. And if it's black, high amounts of mRNA. They've given you 1, 2, 3, 4, 5, 6 genes, and they want to see basically in each type of tissue which genes are being expressed the most, which genes are being expressed the least. Based on the data provided, identify the gene that is most likely to encode a protein that is essential component for glycolysis. Provide reasoning. So this is a 2-part question. Identify and provide reasoning. Glycolysis, remember, is the first part of cellular respiration, right? Glycolysis is necessary for all living things. What I would hope that you would do in this question is you would think, all right, how am I supposed to know that? And then I would hope that you would go ahead and default to, well, I know glycolysis is important for the production of ATP. I would assume that all tissues require the production of ATP. Otherwise, they just wouldn't really function at all. And so what you would need to do is say, which one of these genes is actually active in all tissues? And it would be gene G. So you would identify gene G as the gene that's most likely to encode a protein essential for glycolysis, and your reasoning would be all tissues must provide glycolysis because all tissues must have ATP, and that would be your reasoning. All right, on B, it says the researcher observed that tissues with high levels of gene H did not always have gene H protein. Provide reasoning to explain how tissues with high gene H mRNA can have no gene H protein. So we have tissues that are making mRNA but not making the protein. So I'm going to move here. So this was part A. So if you want to look at that, this is how they worded it. Part B, how can you have mRNA in that tissue but no protein? Here's some of the reasoning. Maybe the mRNA is not exported from the nucleus. Maybe the mRNA is not translated. Maybe there are post-transcriptional modifications that occur that cause it to not make the protein. All right, so hopefully you see here how all of the topics we discussed are kind of geared towards gene expression, which is really important. All right, and that's all I have for you today. I kept it under 25 minutes, which was my goal. Tomorrow I am going to cover gene expression and gene regulation, and I'm going to go over another FRQ tomorrow, and then we'll have a mandatory FRQ on Thursday and a Zoom meeting on Friday. All right, talk to you later.